Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject recombinant DNA technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Today's topic is genomic libraries. So in this you will be learning about what are DNA libraries and how these genomic libraries are constructed and how they are screened, the advantages and limitations of these genomic libraries. Dear learners, let's study about DNA libraries in detail. First, coming to the term genome. So genome, it represents the genetic material present in a cell, particular cell. So the genes which are present in all the chromosomes of that cell, it represents the genome. And the study of this total genetic material is called as genomics. So the whole genome of an organism, if it is fragmented into small pieces and if it is cloned in vectors, that particular collection of all the fragments is called as DNA library, and this is used for various purposes. So DNA library, it is the collection of all the DNA fragments of a particular genome belonging to a specific cell. You know that a book library, it is a collection of books where specific books for various subjects it can be searched. In the same way, researchers use this DNA library to isolate specific genes. So coming to the types of DNA libraries. So these DNA libraries, for what purpose they are used? Means for isolating a specific gene, that is the foreign gene, from the source, you require a library. And for sequencing the DNA and also to study the structure and function of the genes, these DNA libraries, it plays a very important role. And if you see the different types of libraries present, depending upon the source of the starting DNA, the DNA libraries, there are of two types. One is genomic library. It represents the collection of the genomic DNA. And the second one is the cDNA library. It represents the complementary DNA which is present in the, that is it represents the total mRNA population of a cell. So based on the source of the starting DNA, if it is genomic DNA, it is called as genomic library, and if the starting DNA is a complementary DNA, then it is called as cDNA library. So if you see just this example, how this human genomic library is generated, you know that the size of the human genome is 3 into 10 to the power of 9 base pairs. Suppose, let's say that this whole genome is it is fragmented into small pieces of size 50 kb fragments, and that each fragment, if it is cloned in a lambda replacement vector, so if you clone all those 50 fragment size this DNA fragments in this lambda bacteriophage vector, you will be getting nearly 64,000, you require 64,000 phases to hold that size of DNA. That is 64,000 vectors. They represents the entire genetic material of the human DNA. So in that manner, this collection of this particular vectors, they represents the entire human genomic library. That is the 64,000 fast vectors, they represents the human genomic library. So this can be used for, in that manner they can be used for isolating the gene and for studying the structure and function of this human
genes. And the same, that is, if you take the same example, the human genome, if you are using a back vector, so bacterial artificial chromosome, this vector is having the ability to hold a large insert. So up to 300 kb of the foreign genes can be inserted in this back vector. Suppose you take the same example, if you are cutting this human genome into fragments and of size 300 kb, so you will be getting, you will be requiring nearly 11,000 back vectors are required to hold this 300 kb fragments of this particular human genome. So as the insert size, that is the DNA fragment size, increases, the number of clones required, the number of vectors required to hold that gene segment, it decreases. So you require just 11,000 back vectors. They represent the entire human genomic library, and each vector will be holding that is the DNA insert of size 300 kb. So in this manner you can generate a human genomic library by using either the lambda replacement vector or back vector. So let's see about the steps involved in the construction of the genomic library. And you have learned that the genomic library, it represents the collection of genes which represent the entire genome of an organism. So the first step is the isolation of the genomic DNA from the cells. So you will be isolating the complete DNA, and that one is digested with the restriction enzymes, and that is depending upon the that is the vector which you are used to clone, that is small sized fragments is used never if you are selecting a plasmid as the vector, and large sized fragments are taken if you are using these artificial chromosomes as vectors. So depending upon the types of vectors, you will be cutting the DNA into large or small size. The third step is these DNA fragments are ligated in that particular cloning vector, and then you are calling that vector as a recombinant vector, because it is containing the vector genes and also this DNA inserts. So these recombinant vectors, they are introduced into the host cell, and each cloning vector, now it contains different fragments of the genome, so that, and all the, that is these vectors, they represent the genomic library. So on the step 5, all the plasmids in a particular host cell, if you take a plasmid of a particular host cell, it harbored that is, it will be containing the same insert, and as each plasmid it multiplies in the host cell, there will be 50 to 100 copies of the DNA insert in each cell, because the plasmid is having the self replication capacity, and as we are, whenever you introduce into the host cell, along with the plasmid DNA, our DNA insert will also be multiplying, and in each cell you will be having 50 to 100 copies of the DNA insert. In addition, in each transform cell, which is bearing the same cloning vector, will multiply, so there will be million cells in one colony. And because these cells, the transform cells, when you are supplying appropriate media, they will be multiplying, and each colony, it contains million cells, and that colony, it represents the million copies of our DNA insert. And if you see the step 7, the genomic DNA insert, it can be sequenced just if you isolate a particular colony. So from that colony you can isolate the specific gene which is cloned in that particular recombinant vector. And the step 8 is that insert DNA present in each colony, it can be sequenced, and if you that is, if you read the sequence, that is, you can align the sequences using this, because the human genomic library, if you are taking that one, because it is containing a large size genome, you can't read it manually. So by using these computer programs, if you arrange all the sequence, if you align them, and you can, that entire sequence, it represents the genomic DNA. And if the two aligned sequence, that is, if the two sequences they overlap with each other, they represents a continuous segment, and this continuous segment of genome is called as counting, and which incorporate, that is, the sequence, it may be overlapped between two fragments. So this continuous overlapping segments of a genome, like this, you will be getting long countings, they are generated. If you see this picture, so the first step is isolation of the DNA from the cells, and the second step is restriction enzyme digestion. So you will be getting this fragments of DNA from the genome. These fragments are cloned in the plasmid vector. So thus now this vector is becoming a recombinant vector. So the plasmid genes and the DNA inserts, they are present. So these recombinant vector are introduced into the host cell. So each host cell now it carries our DNA fragment. So because here you are taking 3 fragments, 3 host cell, they represent the entire genomic DNA of this particular cell. So when you provide appropriate nutrient conditions and environment, each cell will be multiplying into colonies, that is, you will be getting numerous colonies, and the entire colony, they represent the, that is, it will be having millions of copies of this particular DNA insert. Like this, all the individual plates, they represent these colonies which contain the entire genome of the particular organism. So if you want to sequence the genome, you will be isolating the transformed cells, and you will be extracting the that is the DNA insert from that recombinant vector and sequencing this particular insert. So all the DNA fragments are sequenced by aligning them using the computer program. Sometimes, that is, the sequences, between the sequences, the two sequences, they will be having overlapping base pairs. So that type of sequences, if you see, that is, if you align these overlapping sequences, they are called as countings. So like this, you will be getting a large number of these countings, and when you align them based on that particular this computer program, you will be able to sequence the entire genome of an organism. This the genomic library, it represents thousands of these long countings which can be characterized and assigned to a particular chromosome, and all these chromosomes, they form the detailed physical map of that particular organism.
Clone libraries offered the same advantage of real libraries: orderly access to information. In most clone libraries, the DNA fragments were stored in E. coli. These are bacteria that normally live in our intestines. Each E. coli cell stored a single segment of human DNA and represented a single book of the library. Clone libraries allowed each human fragment to be tracked and easily copied.
So in this table we will see how many number of clones are required to insert the genome of different species. So the vector here taken is cosmid vector, and each cosmid can hold up to 35 kb of the DNA insert. So this is the common vector, cosmid vector, And if you see the E. coli, the size of the E. coli is 4.6 into 10 to the power of 6 base pairs. So if you are cutting this E. coli genome into small pieces, each piece of 35 kb in size, so you require 410 cosmid vectors to hold the entire genome of E. coli. And the next example, yeast, it is having 1.8 into 10 to the power of 7. So this requires 1500 cosmid vectors to hold the entire genome of this yeast Saccharomyces cerevisiae. So each cosmid vector will be containing 35 fragment, 35 kb size fragment of this particular yeast genome. And if you take this Drosophila, it is having 1.2 into 10 to the power of 8. So you require 10,000 cosmid vectors to hold the entire genome of Drosophila. And if you take the rice, the size is 5.7 into 10 to the power of 8. So it requires 49,000 cosmid vectors to hold the rice genome. In the same manner, so 2,74,000 cosmid vectors are required to hold the entire genome, that is 3.2 into 10 to the power of 9 base pair of human genome. So from this table it is clear that as the number of this base pairs are increasing, that is the size of the genome is increasing, so the number of cosmid vectors required is also increasing. Coming to the next slide, screening of libraries. So different methods are employed to identify the recombinant host cells from the population of this non-recombinant cells. So it includes, first one is the antibiotic resistant marker. This is a selectable marker. So when you use this marker, you can easily select the transformed cells as they are antibiotic resistance, whereas non-transformed cells, they are susceptible to this antibiotic. So when you plate the transformation mixture in a medium containing antibiotic, only our transformed cells, that is the recombinant cells, will be having the ability to grow. The second method is blue-white screening. So here the cells, the recombinant and the non-recombinant cells, they appear in two colors. That is the white colonies, they represents the recombinant cells, and the blue colonies, they represents the non-transformed cells. So just by this visual appearance, you can screen the transformed cells. The third method is plaque formation. So the recombinant cells will be having the ability to form plaque formation, that is the clear zones where the particular cells are growing. They represent, this clear zones represent the recombinant cells, whereas the turbid colonies, they represent the non-recombinant cells. Just by seeing picking the plaques, you can screen the recombinant cells. So detailed explanation of these screening methods, so you can go through my previous videos, and in the coming video you will see the animation of another method of screening. So moving on to the advantages of genomic library. So it mainly helps in the isolation of the desired gene from the source. It helps in sequencing the entire genome, and it is helpful to study the structure and function of regulatory sequences under in vitro condition, and it is also helpful to know about the genetic mutation, and it is used for the gene therapy treatment also. Then it is helpful for the identification of the novel pharmaceutical important genes, and it helps to understand the complexity of the genome. Coming to the disadvantages of genomic library. So genomic library is particularly useful for relatively small genomes. Because the size is small, it is easy to construct and screen this genomic libraries in case of the prokaryotic organisms. But in eukaryotes, the genome size is large and it contains a large number of clones, and these clones, sometimes they contain only the non-coding sequences, because the size of non-coding DNA is more than that of the coding DNA, and these untranslated regions, they will be interrupting the coding sequence. So it is difficult to construct this large sized genomes. And another limitation is this prokaryotic organisms, they lack this post translational mechanism. So if the screening method, it requires the expression of this eukaryotic gene in this genomic library, it is difficult. Let's conclude the session by just rewinding some of the main points. So you have learned that the DNA library is the collection of all the sequences from a specific cell, tissue or organism by using suitable vectors, and you have learned the different steps involved in the construction of the genomic libraries. So the genomic library, it represents the collection of the cloned DNA fragments, and it represents the entire genomic DNA. And the screening methods, you have learned about different selectable markers like antibiotic markers, the blue-white screening and plaque formation. And the advantages, you have seen that it is mainly used to study about the structure and function of the non-coding sequences, and also to isolate the desired genes from the source and to sequence the genome. This particular genomic libraries, they are preferred. And in the limitations, you have seen that the eukaryotic genes from genomic libraries could not be expressed in the prokaryotic system due to the lack of splicing mechanism. So with this, I conclude the session.
Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.